Great, perfect. So, welcome to this information seminar for the Knut and Alice Wallenberg Foundation and SciLifeLab Proof of Concept Grant in Life Science. We have scheduled the meeting for today for 90 minutes to allow time for open discussion and questions. We might not fill up that time, but we'll see. At least there's enough time. My name is Isolde Palombo. I'm a project coordinator at the operations office at SciLifeLab. I'm also the program coordinator for the Alpha Cell Research Program. First, some practical info about today. The seminar will start with information about the call, and then we will hear from two researchers that received the grant in 2024 and are now 1 year into the project. There will be a time for questions after each talk, and you can ask questions either by raising your hand or by writing it in the Q&A section here and in the Zoom. There is also on the SciLifeLab website, there's an information page about the call, and there's also a form where you can ask your question. So fill in the form to ask a question, and we will reply to your question. This is the only channel for us to answer question about the grant. So you see that. So no other emailing will be answered. There's also a section with frequently asked questions where we gather questions from the years that we have been having this grant together with Wallenberg. And I think you'll find a lot of information on the website as well. And I think it's also posted now in the chat. Great, perfect. So I think, and also the last thing is the slides with the information will also be shared afterwards. And also this recording will be found on the website. So with that, I want to introduce our first presenters. And we have with us today Professor Olli Kallionniemi, professor at KAW and also SciLifeLab group leader, and the chair of the review panel for this proof of concept grant. We also have Lilian Wikström, who's the director of innovation within the life science at Knut and Alice Wallenberg Foundation, and also responsible for the life science proof of concept call. And with that, I leave over to you.
Okay, thanks so much, Isolde, and sort of good to see everybody for my part as well, and welcome to this POC information webinar. So I will go through quickly a little bit from the point of view of a chair of the evaluation panel, as I have been since the launch of the program, as to what these grants are all about and how do we view them and what they should accomplish, and hopefully that will be advantageous for you to consider when considering whether you would apply for this. So this is to bridge the gap from academic research to innovation. It is meant for scientists who are affiliated with a university to take their discoveries forward. And it's really an academic grant. It's not a business development grant, and it sort of should be something that we all should be thankful to KAW for the fact that they created such a grant program to allow scientists to take their own discoveries forward towards innovations. So I like to often start by, and I should maybe say that we are doing this now for the fourth year. So more or less the same profile has been used before. So what is really proof of concept? The term was actually coined by NASA already in the 1970s, but it's now broadly applied everywhere. And it's really the concept about how we bridge academia to industry and how do we make this bridge that sort of otherwise can be pretty difficult to cross. And that's what the proof of concept grant is on its part. It doesn't do everything, but at least it contributes to the building of the bridge. And I should also like to sort of contrast this to the bridge that we often talk about in translational research, between like a patient and a researcher, that how there's a major gap that needs to be crossed. But this is not what this grant is trying to do. So this is not intended to bridge the entire translational research gap, be it in medical research or be it elsewhere. It is specifically about bringing innovations forward. And it's also not meant for the later stages of helping an already launched business to develop their product forward. So it's really for the academic side of the bridge. So you are like exactly at the point where this guy with the hat is located. And this is open for all fields of life science. But the point is that because this is a KAW funded, KAW driven program, the prerequisite is that people should have prior support from KAW. So you need to be funded at some point earlier on by KAW, or you need to be part of KAW supported strategic research programs. And I'm going to sort of go through them in more detail, but just bear in mind that it's open to all fields of life science and even med tech type of applications that are perhaps not even in this picture. Besides the proof of concept grant that you compete for, there is a process that is called Wallenberg Launchpad, and Lilian will describe that a bit more. But more people will be selected to participate in the whole process, and then only some of them will actually be invited will actually receive the proof of concept grant. So this is a two-stage process in that sense. The eligibility is a little bit complicated, so I just want to run this quickly by you and also explain why these rules. So maybe now let's take the why first. So as a funder of the program, KAW has defined the eligibility to be such that they support research that they have already supported before. Many times at SciLifeLab, we have done it such that KAW gives money to SciLifeLab and SciLifeLab runs the grant. And then we obviously are always keeping it open to everybody. But this is not run by SciLifeLab. This is really a KAW grant that SciLifeLab only helps to evaluate. And therefore, the eligibility is defined by KAW. And first of all, you need to be employed, or the recipient needs to be employed at a Swedish university. You can also have appointments elsewhere, like at a hospital, but the recipient of the grant and the grant should go to the university. And the scientist, the PI, should have support from the Wallenberg Foundation, either as a fellow scholar, clinical scholar, or have a PI or a co-PI in a KAW grant. Alternatively, even if you don't have personal support from the KAW, if you are part of the Wallenberg Centers for Molecular Medicine, if you are part of the DDLS program, if you are part of the SciLifeLab community, meaning SciLifeLab fellows, SciLifeLab group leaders, or infrastructure platform personnel, you can apply. And we have one exception to the rule, is that those who are users or customers of the DDD platform can also apply, and also other Wallenberg strategic programs, but those are seldom relevant to this call. But if you want to apply, there is a point where you can explain, and we need you to explain what is your connection to prior KAW funded research, because that is an eligibility criterion. And just a few points to clarify that just being a user of SciLifeLab infrastructure is not enough, because there are 4,000 projects, so that doesn't make you eligible. But if you have a project accepted in the SciLifeLab Drug Discovery and Development Program, you are eligible to participate per se. We also encourage anybody who is considering to apply with the therapeutic concept, with the therapeutic innovation, to seek guidance from the DDD program. If you don't do that, you are still eligible to apply to this program, but then you need to explain how are you going to cover all the other costs associated with drug discovery, and how do you intend to acquire the expertise that is needed to make drug discoveries or therapeutic drug discoveries go forward. One other tiny little exemption, and I'm sorry that this doesn't apply to everybody, but I'll just sort of quickly say that there's also a question of when do you have sufficient link to a KAW funded research. We made one kind of a borderline exception case that if you are supervising a DDLS funded PhD student, that really doesn't qualify for proper KAW support. But if in that very specific project that the student is engaged with, there is an innovation, you can apply. So these are the rules of the game. And with that, let me describe a little bit based on how have things worked in the prior years. So I'll show you the 2 last years and then last year's sort of a round. So we have between 40 and 60 applications. The review panel that I chair has then typically narrowed it down to about 18, 20 applications that are then invited to participate at the KAW VALP training pitch. And then both years, 10 granted applications came in. So just if you get selected to the first phase does not yet guarantee that you are getting the funding, which is then 1 to 4 million SEK for a 2-year period. Last year, there were more applications. I think there's a little bit of a tendency now for the number of applications to increase. So last year we had 68 applications, 20 accepted, and eventually 11 grants were provided. And as you see, the applicants come from all over Sweden, all different universities. And the topics cover therapeutics, diagnostics, research tools, medtech, industrial biotech, a few e-health applications, and so forth. But as said, you are eligible to bring to us any type of life science sort of a application. So just the very recent grants that were approved are here. They were published just around was it December or something like that. This is one that I'm not going to go through them, but just that you get a little bit of a feeling for the types of projects that were considered fundable out of the last year's applications. And this is just a summary of the past 3 years. So 12 therapeutics applications, 9 diagnostics, 7 technology, 2 research tools, and 1 biomaterials. And as I said, there are more that are actually invited to participate in the process than those who actually get the grant. And just lessons from the prior years, what the panel has usually found, what are the criteria of what the panel usually likes? An exciting discovery, scientifically sound and clear. Clear use case of the innovation, so significant potential, at least theoretically speaking, and an opportunity to develop a clear product. And the product can also mean that you either plan to set up at some point in the future, which doesn't have to be, everything doesn't have to be planned, a startup for it, or it could also go the licensing route, and both are perfectly fine. We require some kind of an external interest or proof of validation to the work that you suggest. And very importantly, that it shouldn't be open-ended, that it should suggest work for a 2-year period that clearly will have an impact and improve the value and validate the innovation, and not be just open-ended thing. And reasons that people have not got the grant have often been that applications were considered too early, that there was a little bit difficult to come up with the clear idea of when and in what way this would be useful, and how would a 2-year grant help that. Unclear value, unclear sort of end users, and not clearly defined added value. And then there were some applications that were already uh, sort of a translated to a company setting, and intellectual property was not in the hands of the scientist, and therefore we can't give the grant to a company. So this is an academic grant after all. So some were thrown out because of those types of reasons. And again, the roles, this is really a KAW program. I just want to emphasize that in that sense. Said that many times. What SciLifeLab role is to help to announce and propagate the proof of concept call, contribute to the evaluation panel, and also announce the grants. But this is not a SciLifeLab or government-funded program, and therefore that's the restriction of the applicants. KAW will make all decisions. SciLifeLab is not involved in the decision making here. And with that, just to point out, the call is open. All the information is available. Call closes in 20 days, so you still have some time to do it. On the SciLifeLab page, you will have more information. And there's also a frequently asked questions that concern, for instance, the eligibility issues. That study those. And with that, I'm done, and I will pass it over to Lilian.
Okay, thank you very much, Olli. I will. Sure, can you see my slides now?
Yes.
Yes, okay, excellent. Thank you very much, Olli, for this information about the call. I will now go on to give you even more information. But first of all, I'd like to briefly introduce the Knut and Alice Wallenberg Foundation for those of you who are not familiar with it. And then I will go on to explain some more details about the call. Some of my slides may be information heavy. I will not go through them all in great detail, but as Isolde said, you will get the slides after the presentations, and also the call homepage has lots of useful information. Let's see, yeah, here we go. So many of you probably have heard about the Wallenberg Foundations, and the largest one of them, the Knut and Alice Wallenberg Foundation, or KAW as we call it. It's one of the largest private funders of research, not only in Sweden, but even in the whole of Europe, focusing on medicine, technology, and natural sciences. And last year, the funding was quite substantial. It was 2.5 billion Swedish kronor. So this is a very important program for Sweden. And the funding to the research comes in through the strategic grants or different types of programs to excellent individuals and projects. So among the grants, we can note the individual grants that you probably have heard about, the Academy Fellows and Scholars and Clinical Fellows, scholarships, and so on. There are projects. There are strategic grants. Olli mentioned a few of these, including DDLS and WCMM, and so on. And then the newcomer in this list of grants is the proof of concept grant that really has come into action during the last few years and fits very well with the aim of the foundation, which really is to be landsgagnelig, the Swedish word for being useful for Swedish society. So the aim is really on strengthening scientific, free, basic research with a goal of benefiting society. And for this reason, now the proof of concept grant has been added to this portfolio to provide the researchers with a possibility to test if their research results could really be the basis for developing a product or service that would be useful for society. And many times, if the answer would be yes, then the idea or the product needs to be commercialized somehow. It can be done through licensing to existing companies. It can also be done through starting a new company. And for this reason, also the foundation has started an investment company called Navigare Ventures. And if you receive the proof of concept grant, There is also a possibility to engage with the Navigare Ventures, apply for funding from there. And the goal again of Navigare Ventures, as of all the other programs, is to give back to society. So if there is a profit, it will go back to this Wallenberg ecosystem. So a few words in general about the proof of concept grants. We have three different proof of concept grants available today. These are distributed through annual calls. The one in life science that we're talking about now. We also have one in quantum technologies, AI, and ICT, which is also open right now. And there is a material science call that's open in the fall. Again, these are academic grants, so they go to the university. You can apply for a maximum of 4 million Swedish kronor for a period of 2 years. And this is a brief view of the process. Right now, the applications are open, and the scientific expert panel headed by Olli will select the scientifically best and most prominent applications, and will be selected for coaching in the Wallenberg Launchpad first phase. And this is when the individual coaching takes place. And some of the projects that have been coached will be then selected for a final pitch and a grant decision, which is when the real execution phase of the project starts for a maximum of 2 years. And during those 2 years, the foundation, we're there for the projects if needed and do what we can to help. And there is also some regular interactions through midterm and final presentations. So to be successful with your application, it's really worthwhile reading all the instructions, because these are not necessarily your typical research grants. Because the idea here is to really find solutions to important problems. So I urge you to look at this in detail. One important thing that people often are not or don't really take very seriously is the verification of interest from external parties. But please consider this. It's not something just a sort of just a rubber stamp, but we're really interested in knowing whether you have verified that the problem you're going to solve is an important one, that there is an interest from the market. So this is not, it's not enough to have a statement of interest from your own startup, but it really needs to be from somebody who's mature in the field. And if you need help with that, it's very good to contact your innovation office. They will be very happy to help you with that, and also understanding the market and so on. Yeah. Some more practical details. All of this can be found on the homepage, but I think the point I would like to bring up here is that the costs that you will ask for, or the money, can only be used for things that money at the university can be used for. So this does not cover patent applications or business development or market analysis or regulatory work and so on. So that's just important to keep in mind when you're planning your projects. Yeah, so as Olli said, it's open now. We have now, in contrast to previous years, tried to shorten the timeline of the whole project cycle. So it's open only until February 20, and then we're going to do all the coaching in May and June, aiming at the final pitch during the last week of June, so the week after midsummer. And then there will be decisions made already in July. So we're trying to adapt to the fast pace of innovation here. So I hope that you think you have an interesting idea or research results that you would like to try to translate towards products or services. And then if that's the case, I suggest you start by trying to define the goal and then the steps that are required to take you there. As quickly as possible, you need to engage with external parties to get the verification of interest. And this may also take some time. But again, the Innovation Office can be helpful here. And I think any other questions that you may have can easily be found at the Q&A on the project homepage. Or you may ask them now because we have some time for questions, right?
Yes. So thank you, Lilian, and thank you, Olli, for this excellent overview. And now, if there are any questions about the scope of the grant or the practical information, please ask them now, or if you want, you can also save it for the open discussion in the end. But are there any questions now? No? Okay. Very clear then. Good. So now to make maybe things a little bit easier to grasp also, we thought it would be valuable to hear from someone who's actually gone through the process of applying for the grant and then also received the funding for this proof of concept project. And first out, we have Marie Jansson. And Marie is an associate professor in vascular biology at Karolinska Institutet and Uppsala University. She has worked on understanding the mechanisms of kidney disease for more than 25 years, and especially then what role the blood vessels have in disease progression and also then potential treatment targets. So please, Marie, stage is yours.
Thank you, and thank you for inviting me to this session today. I'm very happy to share how good experience this whole process was for us. So as alluded to, our project is therapeutic intervention to slow progression of kidney disease. And I will start by giving a little bit of introduction to our project, and then I will kind of move over to why we thought there was a need for a proof of concept project following this, and the benefits we have. We're 1 year into the project time, but already we can see really good benefits from this grant. So I will start by introducing chronic kidney disease. So if we were to summarize that in 3 words, it would be that it's common, serious, and very expensive. So it's affecting 850 million people globally. It is expected to become the 5th cause of death in 2040. And it's also adding to over 8% of our healthcare budget in the European Union. So affecting all aspects of our society. And chronic kidney disease can arise from many different causes. And you can see here on the graph that it's also increasing. So over the last 30 years, it's increased a lot. And the main causes are diabetes type 2 and hypertension, and they account for about 75% of the new cases. But independent of background disease, there are some common features of chronic kidney disease. And these are you lose blood vessels in the kidney, so the smaller capillaries, their density is decreased. You get fibrosis, so this is scar tissue. And once you get this, you cannot, at least not with today's treatment, go back. So this tissue is kind of lost. And chronic kidney disease is also associated with other cardiovascular disease. And we also know that kidney and other organs are communicating. So if you have kidney disease, there's also an increased risk of, for example, cardiac disease. So in our project, it was very important early on to define what was the unmet medical need. Is there even a use for our innovation? And for me as a scientist, of course, I worked with kidney disease for 25 years, so it's quite obvious for me. But when you're applying for this grant, it's very good that you have to define really what is the unmet medical need. And this is often comparing with the current standard of care. So if we look at this pie chart, we have, I will try to get my pointer here. So we have then already patients that are on what is the current standard of care, and this is antihypertensive medication, and then a fairly new drug that's called SGLT2 inhibitors. So this is a sodium glucose co-transporter inhibitor. So when you're on this drug, you urinate more glucose and you urinate more sodium. So this lowers your blood pressure and lowers your blood glucose. And this is quite efficient, but despite this, a lot of these patients still could do even better. And then among this patient group is also a group that we call rapid progressors. So this is up to 23% of the patients. So despite the standard of care, they are not doing well. And then this drug is not suitable for patients that are at the later stage of kidney disease, because initially when you give it, you have a small decline in kidney function, and then you're afraid that you will push them into renal failure and the need for dialysis. So this is not currently given to later stages of kidney disease. And then we also have kidney disease when you have type 1 diabetes. So here you don't want to give any drugs that is also altering the blood glucose, because they are on a strict glucose control. So we think that the drug we have, which is a humanized antibody, and I will show you a little bit about it, but we think it would be beneficial in all of these segments. So if we look then at kidney disease, so already in the introduction, you heard that I'm interested in blood vessels. So we have really contributed to the understanding of how blood vessels are central in developing kidney disease, and that they are kind of upstream of the formation of scar tissue. And one of the molecules that are interesting in this is the angiopoietin 2. And angiopoietin 2 is strongly upregulated both in patients and in animal models of chronic kidney disease. And our data suggests then that this is a causal driver of kidney disease. So this then raises the question if it's beneficial to target angiopoietin 2 in chronic kidney disease. And for this, we have an anti-angiopoietin 2 antibody that is humanized. And it's, I think at least, a pretty cool antibody. So I will, without getting into too much detail, I will tell you how it's working. So in the normal kidney, in the blood vessels, we have a receptor that's called Tie2. So this is very beneficial for the blood vessels and keeps them healthy. And it's normally activated by angiopoietin 1. And this angiopoietin 1 likes to come in a tetramer, and it pulls together these receptors. And when the receptor is a tetramer, it can be activated and you get all these benefits. But in kidney disease and also some other diseases, angiopoietin 2 is upregulated. And then this likes to come as a dimer. So when it binds the receptor can no longer pull together, forming this tetrameric structure. So then you lose all the benefits you have of this vascular signaling. So the antibody we have is actually an angiopoietin 2 binding antibody, but it's binding two dimers. So you can see here. And when you look at it like this, you can see that it looks quite similar to the structure that we have from angiopoietin 1. So we're forcing angiopoietin 2 to become a tetramer. And by doing so, this complex can then bring together 4, again, Tie2 receptors, and we can restore this signaling. So then we can protect the capillaries, and downstream of this, it's also antifibrotic in the kidney. And some of the things with this that is really cool is that it's angiopoietin 2 dependent. So this means that if the blood vessel is not injured, this will not happen. So there's no Tie2 then. So it's a disease homing mechanism. So this also limits the potential adverse effects. So the first studies we did in preclinical studies is now published in this paper just before Christmas. And already before we published this, we saw that there was a potential for this drug or therapeutic to be beneficial for patients. So we already, before this was published, started working with Uppsala University Innovation. That helped us a lot. So we have done a priority filing of this innovation using it in the indication of chronic kidney disease. And we recently filed a PCT filing as well. And quite early on, with the help of the Innovation Office, we did a target product profile. And this kind of made us think what patient group are interesting and how will we administer the drugs. So there were a lot of questions, and this is kind of the basic for why we needed to do some type of proof of concept project. So I already talked a little bit about the current standard of care. And one important thing for us was to compare our drug to the standard of care that is out there and benchmark against that. We also wanted to see if there was an add-on effect with our drug and with SGLT2 inhibitors. And this is important not only because, of course, the patients could get even better treatment if you add on, but also because for a clinical trial, a lot of patients are already on SGLT2 inhibitors, so it would be difficult to do a clinical trial with our drug without having tested this previously. We also had to think about the route of administration. So normally when you do animal studies, it's very common to use a intraperitoneal administration for biologics. So this is what we did in the previous studies, but now we need to change also thinking about the administration route we want to use in patients, and this then would be subcutaneous. So we also changed that. And so this is the proof of concept project that then fortunately was funded by the proof of concept grant. So we have this model of obstruction-induced kidney disease in mice. And here we wanted to give either one dose of our antibody APTA subcutaneously. And in the groups that received SGLT2 inhibitors, this was dapagliflozin, they received that in the drinking water. And of course, when choosing this model, it's important that there were some previous publications showing that in this model, SGLT2 inhibitors were actually effective. And so there are other publications showing that they do reduce fibrosis up to 40%. So this is a good indication. We want to make sure we use a model that's irrelevant, so we don't underestimate the effect of SGLT2 inhibitors. So we then had these 4 groups, one single APTA group, SGLT2 inhibitor, and then the combination group. And in this case, our vehicle is humanized IgG, just to control for injecting a human antibody in the mice. But we have not seen any problems with this previously. So this project is ongoing, and it's looking good. But already before we started, we defined the expected outcome. So one of them was that we wanted expected APTA efficacy to be comparable or even better than SGLT2 inhibitors. And if this is the case, there would be an added value and a larger market size, as it could be an option to SGLT2 inhibitors then. We also, one of the expected outcomes was also that APTA has an add-on effect with SGLT2 inhibitors. And this, we thought, because they have different mechanisms. So we will see if this is the case. But then, of course, there is an added value through a much larger market size. As I said, these are ongoing, but I just wanted to share something about the process of applying for this grant, which I did together with Christer Betzoldts, who's a professor also at KAW and Uppsala University. And I think already writing the proposal, and also you could see that a little bit in the introduction, it was a learning experience, because here you already have to step out a little bit from your academic shoes and get into more of the industry thinking. What are the key experiments, and think about those things. And it's it's a very good thing to start thinking about that and writing it down on paper. And then, of course, we have the coaching phase and also getting funded. And I will go a little bit more into the coaching and benefits of the funding. So I think the coaching was really what you call it. It was coaching. It did not feel like an evaluation, although that was what you expected the first time you met the coaches. They have a lot of experience, and it's very important to appreciate that and also try to understand what it is they don't understand so you can explain it better. They also have an extensive network. Already when I met Lilian after 30 minutes, I told her that we had problem contacting a big company. She said, "Oh, I know someone at the embassy here. Let's put you in contact with them and see if they can help you." And that worked beautifully. But it's a very humbling experience when it's someone you don't really know that's so eager to help you. And that's amazing. It's also important when you are doing these pitches that you actually are coachable. If there are slides you're showing that you have to explain extensively because it's difficult to understand them, you need to change them because it should be easy to understand your project. So it is important to be coachable. But all in all, I thought this was an amazing experience. And I think we thought already from the beginning that even if we don't get the grant, it was very beneficial to go through this coaching phase. And we were lucky to get this grant. And here I've just listed a few things that I think have come out of the coaching and funding and also hopefully will in the future. So the whole process is kind of a feasibility assessment of your project. Like, is it realistic to think that you can commercialize this and move it forward? For us, it was very important to benchmark against the current standard of care, and in a therapeutic project, this is often the case. I think it was very useful to have to dig really deep into the unmet medical need and what the competition is out there and be able to defend our project in comparison to those. I think it's also important to very early change your mindset a little bit, going from an academic person to thinking a little bit wider. And of course, by getting this funding and being able to do this project, you're also de-risking the project for future funders and partners. You are also much better prepared to talk to investors in the future because you have already pitched your ideas and the weaknesses have been pointed out, and hopefully you can answer them now. And this, of course, will accelerate our project towards toxicology studies. We have also started working on the plans for the first in human studies, which is not happening now, but we have started thinking about that and how to do it. And in our case, we have also started a company, and this was not necessarily because it was needed for driving the project forward. It was more a legal thing because we are in discussion with other companies, and if you need to sign documents, you don't want to do that necessarily as a private person. And by that, I also want to thank, of course, the Knut and Alice Wallenberg Foundation and SciLifeLab for this grant, and especially the coaches I met, which is Lilian, Michael, and Sandor. And of course, Christer, I've already mentioned him, and the people that are working in the lab. And of course, Uppsala University Innovation that has been more or less holding our hand during this whole process. So thank you for listening.
Thank you, Marie, both for a very intriguing research project and also for sharing your story about the proof of concept process. There are already some questions. I just want to remind people that please post your questions in the Q&A. It would be much easier for us here to track Alicia here next to me is trying to also answer questions as we go along. But please try to write them in the Q&A that you find here at the bottom of the Zoom panel. So before we go to the next speaker, I'm going to ask Per to stop recording.